Welcome, MS translators. It is my pleasure to be here to do another one of our recorded interviews covering MS Virtual 2020. And it is with great pleasure today that I can introduce Mary Horton. Mary's from the University of California, Berkeley. And it's exciting to have her join us today because Mary actually received the Best Young Investigator Oral Presentation Award. Just to embarrass you really early in by mentioning all of your achievements. At the MS Virtual 2020 conference, heard her presentation. It was fantastic. So reached out and said we would definitely like to do an interview, and thankfully she accepted. So thank you for joining us today, Mary. Can I ask you maybe to start just by introducing yourself a little bit and telling us sort of in broad terms your general research interests?
Sure. Well, and thank you, Brett, for inviting me. I'm really excited to share some of my research, and I am a young investigator, so it's always fun to interact with as many people as possible. But yeah, as you mentioned, I'm currently at the University of California, Berkeley, where I'm getting my PhD in epidemiology, and really my research interests are all about multiple sclerosis and how kind of your genetic susceptibility and tons of different environmental exposures can all come together. I think we all know that MS is a super complex disease. Probably how people get it are different for each person. Kind of how your disease progresses is very different from person to person. And kind of what explains those complexities involves a lot of different factors. So very broadly, I'm interested in how multiple factors kind of work together. And I think this research about the microbiome is very interesting because I mean, our gut is just full of thousands of different particular bacterial species or fungi species, whatever organisms can colonize the area. And it's also, an interaction place for your diet. It's an interaction, vitamin D can impact your gut. MS treatments can impact your gut. Exercise can impact your gut. So it's kind of this melting pot of how all these different exposures that might contribute to getting MS or relapses can kind of be in a certain area, kind of all melting and merging together, which I just think is so interesting.
Yeah, definitely. And it's certainly an area that's of huge interest to the MS community. I mean, we know anytime that we talk about research into the gut microbiome, it does get a lot of engagement and people are very interested in the potential for this. Yeah. Talking about that project specifically then, can you give us a bit more detail about that work?
Sure. So I am part of a project that we're doing. So like I said, I'm at the University of California, Berkeley, and this is done in collaboration with a number of faculty at the University of California, San Francisco, so just across the bay. And the PI there is Emmanuel Wabant, great mentor. And there is a project going on there that started as a pilot study where there are pediatric cases of MS, so people who were diagnosed before they were 18 years old. And kind of over the last few years, they've been collecting stool samples from these individuals to get them recruited into the study. And since, pediatric onset MS is a really rare subset of MS, there's also a lot of other centers, mainly across the US, that are involved in order to kind of get enough samples that we need. And the purpose of this particular study is to see if there are features of our gut microbiome that are associated with relapse. And this is really interesting because, to the best of my knowledge, this is really the only study where we have both information on relapse. So we're like following people over time. When they come to the clinic, we get information about the medications they're using. It's really the only data set where we can look at the microbiome and relapse. Most other research studies that are about the microbiome and MS are about, does it affect you getting MS? Or now that you have MS, how does my microbiome look compared to somebody else who doesn't have MS? So this is a really unique data set. And I think such an important question to be, it makes sense to wonder if there's something about the gut that can impact actual relapse and disease progression. And this particular study, there are 53 people, which is kind of a modest sample size, but doing any sort of microbiome research is incredibly expensive. So this is actually not small for the field. And in terms of the work that I'm doing to kind of analyze these results, there have been a number of interesting but very preliminary findings, things that haven't yet been peer reviewed, and they were presented at the conference, which I'm happy to kind of share what some of those results are. But it was just like, there was one other pilot study before this, which used the same kind of group at UCSF that we're really building upon here.
Okay, yeah. Do you want to talk more about the background information in terms of, was there a lot known about how this bacteria could be involved in disease progression? I mean, what sort of led to this question?
Yeah, so I think so in the context of wondering whether people who have MS and people who don't have MS have similar or different kind of gut bacterial compositions, there has been increasing evidence that they're different. People who have MS have the environment of your gut looks different. And there have been specific species identified, such as Clostridium perfringens, which is a particular bacteria that in experimental models has been shown it can actually kind of cross your blood-brain barrier, so it can get from your gut to your brain. And how that is possible is a whole kind of subfield of MS microbiome research that's much more on like the animal experimental scale, like thinking about how the immune cells and all those tissues that surround your gut can get kind of leaky and can promote inflammation. And either that inflammatory response can be involved in MS processes, or like actual bacteria can then cross kind of that leaky barrier and end up in your central nervous system. But this is, in its infancy, really. That was one microbe where it was identified, has been kind of experimentally tested. There are multiple observational studies with actual humans where we've kind of seen the impacts of it. But so much more needs to be done, and there can't just be this one microbe, right? And none of this has been confirmed to be associated with relapse, which was just a hugely important area to be investigating, which is really what led to this. And, this particular research is in people who are diagnosed as children, right? But I think the broader implication is that if we can learn something from this data set, we can learn about MS more broadly. We can see if it's the same situation for adults. And there's just so much that can be done. It's so exciting to be involved in research about the microbiome, because it wasn't until the late 1990s that people even knew that it existed or was important. It's like this organ that's just been there, and it makes no sense that it wouldn't somehow be involved in health.
Yep. Yeah, I mean, I'm not going to show my age and talk about what actual time I first started doing MS research, because we don't need to reveal how old I am. But back then, which isn't that long ago, I mean, the gut microbiome and its link to MS was basically not talked about at all. And now it's, in the past 5 to 10 years, and really probably the last 5, it's become such a huge area of research. And as you said, that the gut-brain axis and all that, we're starting to learn about that. It is such an interesting area and promising area of research.
Yeah, and it's so complicated because, I mean, your gut, like, imagine all these microbes like interacting with each other. Like no one thing is working on its own. And it's so hard to like investigate and try to understand all of those little connections and interactions and how they could be kind of causally affecting MS. It's like so tricky, but so interesting and so important.
Well, and especially, I guess, even trickier when you're looking at a disease environment that may have been around for a while and changes that might have been caused by that. Hence the benefits of studying this in pediatric MS, I'm guessing, where you can at least get quite early on and try and focus more on what the link might be.
That's absolutely one of the benefits of kind of using pediatric MS as a model, is because you're assuming that kind of those disease processes are starting much earlier. In MS, we don't really know what the latent period is. Like, when do you have your first kind of biological onset versus your first symptom? And we think that in kids, that's a much shorter window. So it's easier to study. It hopefully is easier to study some of these really complex environmental interactions.
Yeah. As easy as studying something this complex can be, I guess.
Yeah, nothing's easy.
Yeah, no, definitely not. So I guess you mentioned that there are some preliminary results that you've got so far, and obviously taking into account that you may only be able to share some of them with us at this stage. Can you tell us a little bit about what you've found so far?
Sure. So we took the approach of kind of wanting to identify whether there are specific kind of species of microbes that might be associated, but knowing that, again, one thing on its own probably isn't single-handedly impacting MS, right? So the first thing we did was, are there specific species of bacteria or archaea, whatever organisms? And we found 20 out of, about 2,500 that seem to be associated with relapse. And the top one is called Bladia stercoris, which, when you look in the literature to see what we know about that organism, there's not much known. There was a study that identified it was also associated with lupus, which is really interesting because another autoimmune disorder. But when you start getting at specific organisms, it's kind of hard to pinpoint the mechanisms that might be working there. So we also then were wondering, well, we know organisms don't just live on their own. They live in these complex niches. They probably work together, work against each other. And so we wanted to see if there were clusters, like if essentially there are groups of these different species that kind of go together and might, as a group, really impact relapse. And we identified one group, of which there was another species in this Bladia kind of larger group that was in this cluster. So things that are in the kingdom, phylum, class, order, family, genus Bladia. I've always got to go through the taxonomy hierarchy.
Yeah.
It seems that organisms that are within this Bladia genus may be associated with relapse. Of course, this definitely needs to be followed up. We need to kind of confirm this work in other cohorts. Since this is a unique data set, we really didn't have the option to necessarily kind of confirm these findings, although there is a group at the University of British Columbia, Helen Tremlett, who's similarly working on the gut microbiome, and we're really close collaborators. And so we've been thinking of how we can maybe confirm some of these findings in that kind of cohort. So, Bladia may be an interesting thing to follow up on. We also identified, well, what do these things do? Like, what do these individual organisms do? And there's this kind of prediction pipeline where you put in kind of these species that you have, and it kind of gives you these predictions about what type of molecular pathways or like what types of proteins they might produce. And really interesting, there were like 10 kind of biological pathways that came up as associated with relapse. And there was one pathway that included, there was kind of one broader pathway that included 4 of these sub-pathways that we had found that had to do with methane production, which is, kind of an interesting and quizzical. It's like we don't know much about what methane does for human health, but it seems interesting that 4 of these 10 pathways would all be involved in methane production. So that is definitely a future step. Again, it's just a prediction. So it's like that predictive algorithm, there's always possibility for it to be wrong. So more experimental kind of research and following it up. Just there's so many interesting kind of future areas of research from this type of work when you kind of have a small sample and it's, one of the first or second kind of papers or studies to be able to look at something. I think.
Well, I think that's the nice thing. It's much better to get to the end of a study and go, we have a lot to follow up here, rather than get to the end of the study and go, well,
Yeah.
that was a nice study, but this isn't going anywhere. So, I mean, I guess one of the next questions we had to discuss was future steps for this research, but you've sort of talked about that already in terms of doing some larger studies and following up
Yeah.
in this work.
I think, finding collaborators who also have this type of data, which seems to be really unique, like confirming findings in those kind of separate samples will be really, really important. Observational studies like this in the microbiome world are really great for generating hypotheses, but they absolutely need to be followed up and tested, like in the lab with experiments. If you can kind of make the gut environment of like a mouse be a certain way, and then within the MS kind of model, that could be really informative to see whether these particular microbes that we've potentially identified are actually doing something. Also, definitely, it's like the chicken or the egg with relapse and things that might be associated with it. It's like, we know that there's crosstalk between your gut microbiome and the rest of your body, right? So it's quite possible that when somebody's experiencing a relapse, that is impacting kind of the environment of their gut. And then that is then like feedback looping into future relapses. So I don't think that's necessarily a problem. It's a super interesting question to wonder, like, how are these things kind of cycling and impacting each other? And could any of this be, increasing inflammation, which then could increase the next relapse? So much future work, to be done. I could get on my little hamster wheel of a brain and just keep going and going and going.
Well, hopefully you've kept sort of the next 10 years completely free to be able to follow up all of this work, because there's a lot to do. I guess thinking a bit more into the future, so we're talking about the future steps for this research, but what are the potential outcomes of work like this for people living with MS?
Yeah. So I think that, some of those outcomes that we want they're long way down the road.
Yep.
But I do think if we can identify either specific species that reduce risk of relapse, like certainly those would be, great kind of targets for some sort of probiotic or some sort of, other kind of transplant, kind of fecal transplant. If we identify, specific species that are actually increasing relapse, there might be some opportunity for like targeted antibiotics to kind of reduce that. But those things are definitely further, like there's so much work that needs to be done to get there. But I think those are the ultimate goals of like bringing this back to impacting people who have MS.
Yep.
And anything we can learn about it would ultimately have that goal in mind. And even diet, if we identify some of these microbes, that can increase risk, subside largely on carbohydrates, then we can think of diet interventions to kind of like kill them off. We don't give them the food that they want to survive. Or if we identify, they can be targeted with some other technology. I think really the options, there's so many potential options.
Yep. And
it's kind of an easy place to try to modify. Like maybe if we can just ingest something, it can directly interact with our gut.
I mean, understanding that things are still a little while off and there's work to be done, I still think the MS community are going to be very excited about this work and hearing about the potentials of it. What can people with MS do to support your work in any way? Is there anything they can be doing to help out?
That is a great question. I think the biggest thing is that if you're propositioned to be in a microbiome study, you provide your stool. Like, don't let that sampling dissuade you. Like we want it. So I think just being involved in the research, since these are kind of unique and rare data sets right now, we really need we just need more information. We need more data from people with MS. I think that would be the absolute biggest thing right now.
Okay, fantastic. Well, I mean, I know that our community are always looking for opportunities to participate in research. They're highly engaged by it. I'm sure they're going to be incredibly interested in your work and incredibly appreciative of you giving up your time to come and talk to us about it today. I'm sure they will have lots of questions and lots of interest in it. So thank you again for not only coming and doing the interview, but for all of your hard work in this area.
Absolutely. Thank you. It's exciting work to be doing, and happy that it's, ultimately going to help people hopefully down the road.
Okay, fantastic. Thanks, Mary.
Bye.